Welcome to season 4 of the Stronger Than Autoimmune podcast. If you're living with an autoimmune condition, this is your space. I am not just your host. I am an autoimmune warrior like you. Together, we'll explore inspiring stories, expert advice, and simple lifestyle tips that can make a big difference in how we feel. Now, a big shout out to my listeners. This podcast would not be possible without you. If you're enjoying the show, here is how you can support. Rate the podcast on Spotify, leave a review on Apple, or share this episode with a fellow warrior who needs a little hope. I also want to highlight a few incredible organizations making waves in the autoimmune community. Workers with Autoimmune Ailments Alliance, advocating for the rights of students and workers with autoimmune conditions. Learn more or become an advocate at WAAA.org. Brightly Thrive, building a strong community for those with autoimmunity. Check them out at brightlythrive.com. And Leapcure, connecting patients with clinical trials and championing patient advocacy. Learn more at leapcure.com. Now I'm sending you all my love and healing vibes from my heart to yours. Now let's dive in. Hello, autoimmune warriors. Welcome to another episode of the Stronger Than Autoimmune podcast. In today's episode, I have Tyler O'Malley. He is the Associate Vice President of Clinical Affairs at Exagen. Exagen is a leader in autoimmune testing solutions. With over a decade of experience, he oversees clinical trials, bioinformatics, and medical policy development, contributing to advancements in autoimmune diagnostics. Tyler has authored numerous peer-reviewed publications, including one of the largest lupus diagnostic studies, and has played a pivotal role in over a dozen clinical trials. A summa cum laude graduate of Georgia Gwinnett College with a degree in biology and a focus in biochemistry, his expertise spans medical science education, essay development, and clinical research. So, enjoy this episode with Tyler O'Malley. Welcome, Tyler. Thank you so much for joining me on the Stronger Than Autoimmune podcast.
Thanks for having me.
Yeah, I want to start with more of your personal journey up to this point. Your path into the field of autoimmune diagnostics is quite inspirational. What initial spark or interest in biochemistry started you up in the autoimmune research area?
I've always been interested in autoimmunity. It's the area of science that has always fascinated me the most. And, I've just found it interesting in that it's this very complex system with a lot of moving parts that, when it's balanced, when it's working well, it's incredibly powerful, right? It protects us from a wide array of pathogens and things that can make us sick and harm us. But when things go wrong or things become unbalanced, things can go very, very south very quickly, and the body begins to attack itself. And so, understanding the various ways in which that can happen, and ways that we can treat that and manage that, I've always found that just to be a very interesting thing to learn more about.
Yeah. So pretty much you just love to learn and science and fascinated. I'm glad we have somebody like you on our side. And as someone with over a decade of experience, how have you personally or professionally, these experiences you've had, shaped your passion for improving diagnostics with autoimmune diseases?
So I'm still as energized and excited about the work that we're doing here at Exagen as I was, as a young employee learning the ropes. And obviously now I have a lot more resources at my disposal to be able to conduct research and move things forward. But it just comes back to always keeping the patients, the people behind the tubes of the blood that we receive every day, top of mind. Humanizing the work that we're doing, it's important because sometimes when you're working behind a computer or talking about very abstract concepts like we do day to day, you can lose track of that. So that's why I think these sorts of things are important. Engaging with folks that are dealing with this and living it every day.
Yeah. I had my first Exagen test maybe a couple years ago. And for people that aren't familiar with Exagen and autoimmune testing, would you mind talking about what it does? And I mean, it's pretty much just them, y'all get our blood. So it's nothing on our part, but how could you explain what you do on your end?
Sure. So a common reason why patients who have early signs and symptoms of autoimmune disease make their way from a primary care physician to a rheumatologist, the specialist that generally deals with a lot of these autoimmune conditions, is because their primary care physician sees some of these symptoms that maybe you've dealt with, like brain fog, fatigue, arthralgias, just generally not feeling well, right? And oftentimes this will trigger the primary care physician to order some very general tests, like inflammatory markers like CRP, which don't really tell you anything specific about your condition, but it can just tell you that you have an abnormal level of inflammation. Another common test is what's known as ANA, anti-nuclear antibodies. Again, a very general test. It doesn't tell you that you have lupus or another test necessarily, but it does indicate that you're having some level of an autoimmune reaction that's manifesting itself in the form of antibodies that are building in your blood. And that can point to an autoimmune disease. And that's where the rheumatologist will kind of step in and try to learn and understand better the signs and symptoms, connect that to the ANA, and oftentimes this is the entry point for an Avise CTD test, which is one of the main tests that Exagen offers, and it's really meant for that next step for the rheumatologist to put the signs and symptoms of a patient who has these non-specific overlapping signs and symptoms that could be lupus, could be rheumatoid arthritis, could be Sjogren's, could be an overlap of these different conditions. Sometimes they coexist together. And so this test, it consists of 23 biomarkers, which sounds daunting, but the test, it goes through some effort to put this information together into a integrated or consolidated result that brings clarity specifically to the likelihood for lupus. So there's an algorithm that reduces 10 of these biomarkers down into a scale that's interpreted by the rheumatologist. And then you have other biomarkers, many of them antibody tests that can help diagnose other related conditions to lupus and help differentiate those signs and symptoms. So it's really meant to be a roadmap to pick up on that journey after the patient arrives at the rheumatologist with that positive ANA, a point in time which we recognize is probably a point in time in a patient's life, very often a young woman's life, that is probably fraught with a lot of uncertainty, anxiety, and a time where you're looking for answers.
Yeah. So it pretty much gives you more information on which direction to go, or the rheumatologist, and what's going on with your body. I do have a question because, I mean, obviously I think it was maybe it was 3 years ago, but not recent. Does the information change these biomarkers, or how often should you get this test? Since, I mean, talking about me, asking about me.
The simple answer is it can, and they're really, it's interesting. A lot of times scientists or people who work in science, they avoid yes or no questions, but I think it's particularly the case here in autoimmunity because there's very little that's black and white in autoimmunity. It's everything exists on a scale or a gradation. Um, and so, yes, you can have a certain level of an autoimmune reaction at one point in your journey, and you can have a lesser level of an autoimmune reaction at a different point. And oftentimes that's a good indication. However, there are some biomarkers that tend to be more static, that sort of stay still, regardless of whether you're doing well or you're not doing so well. And there are other biomarkers that are more dynamic. So, as an example, anti-double-stranded DNA antibodies are often thought to fluctuate with the disease. So when you're flaring, when things are not going as well, the antibodies tend to go up, and the opposite happens when you're doing well. On the other hand, anti-Smith antibodies, which are very, very specific to lupus, they don't tend to be responsive to these fluctuations in disease. So again, this is a reason why it's important to have a rheumatologist guide you through the results and make sense of them, because some of these details are nuanced.
Yeah. Well, thank you for answering that. Even I got the however and yes and no. It does help. You overseen bioinformatics and clinical trials. How has technology like AI and machine learning transformed the autoimmune space? And what's next?
Yeah, I mean, in a word, tremendously. And really, what it comes down to is it's scale. In that, particularly with bioinformatics, we have capabilities now where we can test different combinations of biomarkers together um, in a much shorter period of time than we ever could before. And so, where something that could have taken us, weeks to do before, we can do it in a matter of days. And this is really important because ultimately, what we're after, and what you're generally after in science, not just here in autoimmunity, is the simple things. You want E equals MC squared. Complex answers usually are not the right ones, in that they tend to fall apart once you better understand the problem. And so that's why it's really important to sort of test out a lot of different combinations of biomarkers so that you can get down to the simplest answer possible. And that's what tends to be durable and to be an accurate solution that works for a lot of people.
It sounds complicated, but yet with, like you said, AI has really made it more simple. I'm imagining, tell me if I'm wrong, but I'm kind of thinking of like almost like a flow chart, yes and no. And it's like, yes, this, and then why this, and, getting deeper and deeper. I mean, if you were to give an image, because I'm very visual, of how this works when you're testing our blood and bioinformatics, can you give us one, or can you give me one?
So are you asking like how the algorithm works, or are you asking like how like development would work, like how we would decide which biomarkers are good? Or
Well, now that you said that, I mean, yeah, could you answer about the biomarkers? I think that's more of an interesting question.
yeah, so okay, so visual, right? So imagine a funnel, right? So we, there's, we do a lot of protein work. We don't do molecular testing here. So we don't do like DNA, RNA stuff. So we do proteins, right? And there's probably, there's about 20,000-ish or so proteins that are characterized. There's probably a lot more, but let's just say 20,000-ish. So, we have the ability now, and this is, again, is, just an example of technological advancement. We have the ability now with a very small amount of blood to be able to measure, let's say 1,200, 1,500 proteins on a given patient sample. So we can run a clinical trial with a group of lupus patients, for example, collect their blood, and then screen all of these patients for 1,500 proteins each, right? So we've got a massive amount of data. Think of an Excel sheet that would, crash your computer essentially. And you take this data, you connect it with the clinical data that we, get from the physicians, the signs and symptoms, the disease activity, the medications they're taking, and you construct some thoughtful machine learning models with some clinical guidance, and you take this funnel and you start to reduce these biomarkers down, and you're creating almost like a ranking system, and you'll end up with a much smaller set of biomarkers. Think maybe 10 to 20 or so, and you develop sort of a leaderboard of performances, and there's different ways of measuring performance characteristics. And so you have this leaderboard, and then once you have a leaderboard and you say, okay, I think these are a set of biomarkers, these 10 biomarkers are very good. Then you go through a process where you try to simplify, where you say, okay, here are my 10 critical biomarkers. I'm going to take the 10th least, the 10th biomarker in this list, the least important. I'm going to try to remove that biomarker and see if my model still holds up. Can I go to 9 biomarkers? Then can I go to 8, and then to 7? And then you see where the model breaks. And this is kind of that process I was getting at earlier, where you're trying to simplify. So that's a process that you just repeat over and over and over and over again. And again, there are tools that make this very simple now and automate a lot of this. But like anything else, it requires, there's a very common, almost, trite saying at this point, garbage in, garbage out with bioinformatics. My role is a little unorthodox in that I kind of combine disparate parts of the organization that are often occupied by different individuals, clinical affairs and bioinformatics. It's useful in a lot of cases because having those two sort of thought processes coming together at the same time is often very useful.
It sounds like you're able to really break down information in a way that's digestible. For people that aren't familiar with, or like we already mentioned, you do blood testing, but you do clinical trials. Are the clinical trials limited to certain things, or how does that work? Because I've only heard of personally the blood testing you do. How does a clinical trials work?
So our focus is primarily on blood testing. There are other potential future areas of expansion. There are devices that companies develop. There are sort of other areas that we could potentially expand into. But right now, diagnostics is blood testing specifically is our area of expertise. And so that's what a lot of our clinical trials focus on. That's not to say that every clinical trial we do looks exactly the same. There are different types of trials. So for example, some studies are retrospective, where, for example, we've gone back and looked at medical records for patients we've already tested and connected the test results that we have to the medical records, and then try to understand, how the test results influenced the physician's behaviors in terms of how they diagnosed and treated the patients in response to those test results, and how that compared to physicians who were relying on, conventional means to diagnose lupus patients. This was a study that I worked on personally and published back in 2022. That we were able to show very significant differences there with our testing. Other studies are prospective, where we're setting up a trial where, for example, we're saying, okay, we're going to take a set of patients that meet a defined set of criteria. We'll give some of these patients the AVISE test. We'll give other patients, the conventional, approach for diagnosing lupus, and then we'll follow these patients out for a period of time and collect outcomes on them. So there are different ways of doing this, and there's reasons for doing the trials slightly differently, but it's never boring. I guess I'll say that.
Well, and that makes sense. I mean, that you're doing clinical trials for different things, because just like you mentioned, going back to, if you have to ask the right question. And if you're not even asking that question,
Right.
well, you're not going to get that answer, or you're not going to get any information on whether that is real or helpful or not for the patient. With these clinical trials, are they advertised, or is it just through the doctors, or how can people really sign up to start working with your company?
So, clinical trials are, governed by, a set of rules, and specifically through what are known as institutional review boards, or IRBs. And so there are ways that we can promote enrollment. So we've done things like this before, where we can create sort of informational flyers and things like this that go through IRB approval and such. So we've done this, and it's proven effective for us. But really, a lot of this relies upon the physician, and very often the physician staff, be that a clinical research coordinator, which a lot of times, frankly, is a nurse or an MA who's sort of wearing two hats or three hats, and taking the time, often between patients or after hours, to comb through medical records and identify patients that would be ideal for trials. And then, the other part that's, very critical and, sometimes overlooked is the process of consent, of really taking the time, trying to help someone understand why they're being approached to participate in a trial, why it's beneficial. It could help other people like them in the future. Their contribution to science, to the greater good, and ultimately getting the patient to consent to participate. It's not a given, and not everyone does participate,
Yeah.
and it's not as easy in some groups as it is in others, as has been very well documented.
You do a lot of work in the lupus studies. What findings surprised you the most, or most impactful for patient care?
It's an interesting question. I'm always surprised that, in lupus, the disparities are as big as they are, and they continue to be. There's been, tons of ink spilled on this question of, is it genetic? Is it a byproduct of socioeconomic status, of social determinants of health? Is it some combination of the three? I think I tend to think it's the latter. It's some combination, but that's the thing that continues to shock the conscience for me, is that we have some people that are suffering to such a disproportionate effect, and we don't seem to be getting much better at it, and that's a little disappointing. At the end of the day, I want everyone to benefit from what we're doing with our testing, from new drugs that are getting developed, I would hope that as some of that happens, that we start to see some of those disparities close a bit.
Yeah, thank you. Just like you mentioned at the beginning, autoimmune diseases are notoriously complex. Exagen uses reliable information, but what are the different ranges of the conditions? I mean, is it all autoimmune diseases, or do you all focus on a certain one? You've been talking a lot about lupus, and you mentioned Sjogren's. Any other rare autoimmune diseases do you focus on?
So, yeah, good question. So the AVISE CTD test itself, across those 23 biomarkers I mentioned, addresses 8 different diseases. So you have, we're going to test my knowledge, see if I can remember them all. Lupus, rheumatoid arthritis, Sjogren's, myositis, antiphospholipid syndrome, autoimmune thyroid disease, mixed connective tissue disease, and scleroderma.
You got it. Ding, 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 ding.
Yeah. So, and they're very intentional, right? So it's not that we just sort of grabbed 8 different diseases and put them together in a panel of biomarkers. The thread between them is that they're all ANA associated.
Okay.
So again, it comes back to the clinical scenario, the entry point for the test, which again goes back to the conversation we were just talking about in terms of really thinking about the question that you're trying to answer. I think about what we do is and what we provide. You know, as a company, we're different, right? We don't make a product. We make a product, but we don't make furniture. We don't make soda. We don't make a commodity.
It's more of a service.
Well, I think of what we make, what we provide, or what we sell, if you want to say it that way, is information. And ideally, if we're doing our jobs, we're providing better information than what our other contemporaries are providing to the same customer, the rheumatologist. And so that's what we're doing with our test. To your question about sort of the broader scope of autoimmune disease, there are over 100. If you look at sort of the NIH, there's over probably there's probably close to, I think, 50 million people who have some form of autoimmune disease. So it's a massive category of diseases, and it goes much broader than rheumatology. So, for example, MS, multiple sclerosis, is an autoimmune disease, but it's something that's dealt with by neurologists. So autoimmune is a very much a catch-all basket that encompasses a lot of different things. And again, goes back to what I was saying before. Your immune system is a tremendously complex thing, and it can go wrong in a variety of different ways that manifests itself in all these different diseases.
Will y'all expand to neurology or other parts of the autoimmune system, or you're just going to hone in on what's going on now with those 8?
Want to give away state secrets?
Darn. Okay. All right.
No, no. So of course, I mean, the goal ultimately is to provide clarity where the need is the greatest. So this is something we continuously do, is we try to engage with our customers and engage more broadly than that as well, beyond rheumatologists, to understand where the needs are. I think it sounds simple, but going where the needs are, and that's usually the best place to be.
Yeah.
Some people chase the interesting science. Some people chase what's easy. Some people chase what they think will make them the most money in a short period of time. For me, I think you go after where the need is the greatest, and you figure out a way that you can address that with good science, and the rest of it will take care of itself for the most part.
Yeah. That says a lot about your company. With the clinical trials and research, you've kind of mentioned this about inclusivity, diversity. How do you make sure that this is part of your system, your clinical trials, despite all these different autoimmune diseases and how they're diagnosed?
It's not easy in that as oftentimes as the sponsor of trials, we can't directly control who's being enrolled. So, you know, we can't control, who consents to studies and things of this nature. What we can control are, the investigators that we work with and the sites that we choose, which is oftentimes a strategic choice. There are, certain investigators that are really leaders in this regard at the forefront. So, for example, Dr. Rosalind Ramsey Goldman at Northwestern University, she's really been a thought leader here in terms of um, spending the time working with community leaders in Chicago and finding ways to engage with minority populations to, dispel, you know, um, misunderstandings and things like this about clinical trials, and to get folks who traditionally wouldn't engage with clinical trials to want to engage and understand the value of doing so.
Yeah. Since you are very strategic and you deal with your part of the clinical, you're vice president of clinical affairs, what strategies, how do you bridge the gap between research findings and clinical practice?
It's a process. It's a lengthy process. You can almost liken it to drug development. It's not quite as onerous, but it's close. And um, again, what it comes down to is you really have to think through what we talk about internally, a term that may not mean a lot to um, a general audience, but what we call clinical utility, which is um, you know in simple terms, it's the usefulness of a test. It's not the accuracy of a test. It's not the reliableness of a test. That's actually addressed in a separate set of studies that you also have to do. This is sort of the last stage, the last phase of it, where once you've demonstrated you have a test that's reliable, you can measure what you're saying you're measuring consistently over time. You've demonstrated that your test is accurate. You're actually measuring what you think you are, and hopefully better than whatever's currently available. The last thing you have to do is you have to say, okay, now we have this test. We're putting it in the hands of the physicians that we intend to use the test. And we've shown that when the physicians apply the test in the clinical scenario that it's intended to be used in, that the physician does something with the test results. And that could be a variety of different things, but they do something with those test results that affect the outcomes for the patients in a way that benefits the patients. And again, that could be defined in a variety of different ways. Getting them a diagnosis sooner, reducing their symptoms, preventing them from, a relevant example in lupus is maybe, preventing them from having to take steroids for as long. So, very broadly, that's sort of, that's the test ultimately for any test or any diagnostic test to be viable is whether or not a clinician will take that test and do something with it, and that it'll actually benefit the patient.
Yeah. Yeah. Because, I mean, you could test all you want, but if it doesn't do anything in the long run, but that doesn't make sense. And you mentioned you primarily, I mean, obviously you work directly with a physician. How do you work or give them updated education on with physicians, doctors, nurse practitioners, PAs on what y'all are doing?
Yeah, so we have a variety of different educational channels. So, I'm lucky I have a team of folks around me. So, for example, we have a medical science liaison on my team. She is someone who will travel with our sales team across the country and um, engage in speaker programs with clinicians where she'll take the time to walk through information, educating physicians on our biomarkers, helping them to understand what we're measuring, why we're measuring it, answer their questions and help them understand the test. We actively participate in academic conferences and present scientific abstracts, posters, things of this nature. Um, we're active with community rheumatology groups, as well as patient advocacy groups, a variety of different groups through our marketing group here. So we have an array of efforts to get the message out there and educate folks. And this is something that's very top of mind for me at the moment, because we have just recently updated our AVISE CTD test to include some new biomarkers specifically to enhance our ability to diagnose lupus and rheumatoid arthritis. So very excited.
Yay! So does that mean I have to retest?
Well, I would say you should talk to your doctor about that.
Okay. I had a feeling you were going to say that. All right. So let's move on to misconceptions about autoimmune diseases. Any you can dispel?
Well, to be honest, I don't know how much of a misconception this is, but for the most part, they're not contagious. For whatever that's worth. I don't know if many people have that misperception, but they're generally not. I think I think a misperception maybe that's common in lupus is that the effect of the disease is maybe underappreciated, in that because lupus is a disease that sort of waxes and wanes. You can have periods of time where you feel generally okay. That maybe the perception is, well, are you really that sick, and is it really that bad? Things of this nature. And that's true of other conditions too, to some extent, like rheumatoid arthritis, especially if you're, relatively well managed and all of that, you can be doing relatively well. It doesn't mean that you still don't have pain, or you don't have morning stiffness where, for the first half an hour of your day in the morning, your joints are very tight and uncomfortable. So, I think, inflammation generally is somewhat silent. It's not visible in the way that maybe a rash is, or a physical injury is. So I think that's probably a misperception.
Yeah, I definitely would agree. If you had a crystal ball, anything that you foresee happening, or if that's not an interesting question for you, what about this? If you had your own genie in a bottle or whatever, what would you like to see in the autoimmune space or Exagen?
Yeah, it's a good question. I can actually put it relatively simply. I'd like to see rheumatology go the way of oncology. And what I mean by that is, in rheumatology, in the past 2-3 decades, in the way of drug development, we've gotten very sophisticated in the way that we can be very precise in targeting pathways that get dysregulated or unbalanced in the immune system. Yet we're not precise at all in how we administer these drugs.
Mm.
So, for example, with rheumatoid arthritis, there's 5 major mechanisms of action that are, FDA approved for treatment of RA. Most people get anti-TNFs. Why? Because that's at the top of the formulary for most payers, not because most people respond to anti-TNFs. So you look at oncology is in contrast, and if you watch any amount of TV, you've seen commercials for companion diagnostics where if you have a HER2 mutation, or you have insert gene mutation here, you should take this specific form of chemotherapy.
Gotcha.
So this comes down to aligning diagnostics and treatments together such that we can actually realize personalized medicine. It's one of these things where it's become this buzzword where it's almost people almost roll their eyes now when people say this, because it gets a lot of lip service, but it's very hard for people to point to an example of it in this space. And the way that you get there is by truly aligning the treatment and management of an individual to the individual rather than to their disease. And we haven't quite got there yet in rheumatology.
Yeah, I think that's quite interesting. I mean, I got to admit, being tested like that and finding out what's going on with my body with cancer, having one of those gene tests, and it is a personalized medicine, and I think it's become a word like that because nobody's actually doing it, like you said. Nobody, I mean, besides cancer, I mean, nobody's doing that in primary care. Nobody's really doing that in other realms of health. So, what advice, moving on to your personal insights, what would you tell young scientists that are interested in autoimmune research and diagnostics?
It's an interesting journey, and I think you have to be prepared for the unexpected, and to really, I think the key to staying engaged and not losing sight of why you're doing it is to just stay curious, to remember why you're doing it, to want to continue to understand the problem, to question whether you really do understand the problem, and question your assumptions as you're going along the way. But I think, the reality is, what I've learned is it's very true what people say in terms of the more you know, the more you realize you don't know anything, and I think that's particularly true here, and I think that's, in some ways, it's liberating, because to become an expert, or to understand something at a level that most people don't, and to be able to channel that into something positive, that maybe helps some people somewhere that you'll never meet. You'll never understand the impact. You'll never, feel the joy that it causes them or their family. That means something. I think about that sometimes, and you've got to find ways to keep going, because it's not. Nobody said this job was ever going to be glamorous, and I can tell you some days it isn't. But you've got to find a way to keep going.
Yeah, thank you. I think what you're doing is very helpful. You have that capacity to see things a lot differently from any other person out there. I think every job that people do, really, it does take a special person to do it. So I appreciate your work. What has been the most fulfilling part of your career, and how are you fulfilling it at Exagen?
Well, I have to say, definitely the most fulfilling part has been the past 2.5 years or so, which um, really just includes the time in which I've been managing people. So, for a long time, I was an individual contributor and very focused on myself, my own development, and honing my own skills and getting better. And now having a team of 10 now, um, it's it's a totally different perspective. You have to completely change the way you look at things, um, and you have to understand different personalities and adapt to different folks. But again, it's this giving yourself over to something bigger than yourself, and trying to um, you know help people grow and develop, and hope that has a lasting impact for them, um, and and all the while trying to manage all the you know business objectives and everything else that you have to do along the way. It's always there in the background, but balancing the human element there as well. So um, that's been a lot of fun. It definitely allows you to do a lot more, but it also demands a lot more of you. And it's not just it changes the way that you view success and failure as well, which I've experienced both. The wins are bigger, and the losses are worse, because you're not just winning for yourself anymore, and you don't just lose for yourself anymore either. So um yeah, it's been inspiring to be able to lead people, and I really enjoy it.
Wonderful. And we will close with this patient, with this last, we will close with this last question. Obviously, it's about patients, Freudian slip there. How can patients and providers stay connected with Exagen and learn more about the latest research, solutions that your company is working on?
Yeah. I'm not the best at this myself, admittedly, because I'm a little bit anti social media and such, but I'm told we do have social medias, uh, all of the common ones, and we do maintain a web page that has all of our publications on there, and we have LinkedIn and all those sorts of things. So um, we do try to get our information out there as much as possible. For those that are really interested in the research, the American College of Rheumatology website is a really good one. You can look for all of our abstracts by searching for Exagen. It actually does a pretty good job of pulling all that together relatively easily. So there's a variety of different ways to find us, and if you really want to find me, I'm sure there's a way too. Yeah.
Thank you so much. I appreciate your time.
No worries. It was great. Thank you.
Guess who I just connected with, Warriors? Leapcure. Leap what? Leapcure. Leapcure connects patients with clinical trials to make research more equitable and efficient. Listen to their latest clinical trial.
If you've been diagnosed with thyroid eye disease and are seeking potential new treatment options, you might be interested in the LIDS clinical trial. If you're 18 and over, have a diagnosis of Graves' disease or Hashimoto's, and are interested in potentially participating in research, you can fill out a short questionnaire at lpcur.com/TED. You'll be connected with someone on the team to determine whether it might be a good fit for you. Check your eligibility and learn more at lpcur.com/TED.